Okay, today we are going to study polymerase chain reaction. Okay, nothing but in vitro DNA replication. What is meant by in vitro? In vitro is nothing but outside the body. Okay, outside the body. So DNA replication occurs inside the body normally by using various types of enzymes, but here we are doing DNA replication outside the body by using a machine. Okay, a electric machine which is discovered by Carey Mullis. The scientist name is Carey Mullis in 1985. He discovered this technique, PCR, polymerase chain reaction. By using this technique, we are doing replication of DNA outside the body, okay, or desired quantity of DNA molecules. Desired quantity. If you want 1000 DNA molecules, you can by using a single principal DNA molecule. So where this technique is used? In forensic science. In forensic science. Okay, criminal sites having some DNA samples from blood stains, root hair, okay, nails, body scars, tissue, cells, etc. So the scientists or the criminal sites having such a DNA, we are taking that DNA molecules to the laboratory and we are multiplying the DNA molecule for the further investigation. Okay, so this technique discovered by Carey Mullis in 1985. Only three steps are there in this technique. Three steps. Okay. Step number one is known as denaturation. Denaturation. Denaturation, breaking down of DNA molecule. Okay. So it's a simple machine. First of all, by using Eppendorf tube. Eppendorf tube is a tube, plastic tube. Okay. What we have to do, that DNA molecule you have to put in the Eppendorf tube, which is inside the machine. In that tube, your desired DNA molecule is there. Okay. Then you have to mix various types of enzymes which are used for DNA replication, like DNA helicase, like SSB proteins, like RNA primer. Clear? But except one enzyme, that is DNA polymerase 3. Okay, DNA polymerase 3, which is actually working only in vivo condition, not in vitro. So for in vitro, we are using Taq DNA polymerase. Taq DNA polymerase, that is Thermus aquaticus bacteria. We are extracting the DNA polymerase from Thermus aquaticus and then we are putting that Thermus aquaticus bacterial enzyme in the Eppendorf tube for polymerization purpose. So first of all, the step one is denaturation. Denaturation means breaking down of DNA molecule into two different strands. Consider the original DNA molecule. Suppose this is your original DNA molecule having different nitrogen bases like adenine, thymine, cytosine, guanine. Here thymine, adenine, guanine, and such like that.